Good afternoon. Just out of curiosity, how many of you are still recovering from a physics exam today? Okay. All right, so we transition today to a new topic, the respiratory system. And that's what we're going to do this week. So we'll have 4 classes, with two double classes on Monday and Wednesday, doing the respiratory system. Then we'll have a lab with some respiratory anatomy, both pig and human. And then we will have the exam on cardiovascular and respiratory systems on Monday. So we have an exam coming up. So nothing really of content here other than, well, let's do some anatomy. Well, no, let's start with some physiology after all. So here's a table from your textbook. It includes a few more functions of the respiratory system that we will not be dwelling on. The 3 functions that we are going to focus on are providing oxygen. Now you should be thinking cell phys, why do we need oxygen? What you learned in cell phys, why do we aerobic organisms require a respiratory system in order to get oxygen into our bodies? Well, some of you might remember that oxygen is the final electron acceptor in the oxidative phosphorylation chain. You know, after glycolysis and the Krebs cycle and the electron transport chain, those high energy electrons from NADH have to go somewhere, and they go to oxygen. So we have evolved a system for the use of oxygen as a final electron acceptor so that we can produce ATP by oxidative phosphorylation, which is highly efficient and produces the ATP that we need. We get rid of carbon dioxide with our respiratory system. Well, where does carbon dioxide come from? Again, cell phys. It comes from the metabolism of our fuel, from glycolysis. Well, no, glycolysis does not generate CO2. What generates CO2 is the Krebs cycle. So if we go into the Krebs cycle, CO2 comes out. So again, the oxidation of fuel, carbohydrates, fats, and protein, it turns out, all generate carbon dioxide, and we need to eliminate it, and we do that by a respiratory system. So those things you know, that's sort of intuitive, you know, learned that in middle school. What you may not have learned is that the respiratory system is essential for maintaining the pH, maintaining the optimum pH of your body. So we will get into how carbon dioxide and bicarbonate ion work together to buffer the pH of your body. We're going to talk about that. And those of you who have taken or are taking biochemistry, this will synergize with some of the biochemistry that you're learning. So here is a slide that does some anatomy. There is a bunch of respiratory anatomy in the head. The upper respiratory system is full of anatomical terms, which you are not responsible for. So don't start annotating your slide with this, because I am not going to ask you to memorize the anatomy of the nose, mouth, and upper throat. But I just want to put this slide up there to, to sort of inform you that if you do take anatomy in a medical, nursing, PT program, there will be a lot of anatomy that I don't cover. Just heads up, that's all. Here's the anatomy that we will cover, from the larynx down. We are not covering the pharynx and the nasopharynx and all the stuff up there. So I could go through this slide, but I would, well, what are some of the key points here that I want you to either highlight or emphasize? I mean, all the terms on this slide are fair game for respiratory anatomy. And since you are not having a respiratory anatomy quiz, it could show up on the exam. So the larynx in the throat is where the vocal cords are, and it is part of the upper airway. Trachea is a tube for bringing air into the lungs. The trachea divides into two main bronchi. So one bronchus, two bronchi. There's a left and a right. The main bronchus, each main bronchus, divides into other bronchi. So the main bronchus is the largest tube going into the lung after dividing from the trachea. Then there are bronchi, and we'll be getting into the smaller tubes later in just a few moments. The lung has an apex and a base. The lungs have lobes. So the left lung typically has two lobes, an upper lobe and a lower lobe. The right lung typically, not always, but typically has three lobes, upper, middle, and lower. So I hope you're annotating your slide with these additional bits of anatomy. The things that separate lobes are called fissures. So both lungs have an oblique fissure. This one here, the lowest one, is the oblique fissure. And typically the right lung has a horizontal fissure. I keep saying typically because there is some person-to-person anatomical variation. Right.
And the fissures just like superficial or
They go pretty deep. You'll see on the cadaver on Thursday or Friday that they make a pretty big separation between the lobes. The lobes are connected with each other, but the fissures go a couple inches deep. So it's not just a superficial line. It's a crevasse between the two lobes. So I will continue to use these anatomical terms, but I'll probably use it mostly in a physiological context, except when we're in lab, when we're just going to focus on anatomy. So there's some language that we're getting in common. Here's some language that we're not going to be getting in common. We will be talking about the esophagus, but not until we get to the digestive tract. Trying to see if there's any language on this slide that I will expect you to know, that I would ask you to write down. Other than esophagus, which is digestive, I'm not sure there's anything there. If I offer extra credit, then this slide could be available for extra credit labeling if I give you the head. Obviously, if I don't give you the head, I'm saying I don't want to see any of those labels. So now let's look at the lung airway. Let's focus on the airway of the lungs. Trachea, right main bronchus. Bronchi, major tubes branching from the main bronchus. They branch and they branch and they branch and they branch till we get to microscopic airway tubes called bronchioles. So sort of like arterioles, our arteries divide and divide and divide until we get to arterioles. Same thing with airway. Bronchi divides and divides and divides and divides until we get thousands of bronchioles. And at the end of the bronchioles are the respiratory alveoli. Alveoli is where most of the gas exchange is going to happen. So functionally, alveoli is where we get our oxygen and get rid of our CO2. This slide shows the dividing. So we have one main bronchus, one trachea, two main bronchi that divide and divide and divide until we get to bronchioles. We've got something like 60,000 bronchioles that divide and divide and divide until we have something like 8 million alveoli at the bottom. You don't have to, you're not responsible for this slide. You're not responsible for the number of divisions. You're not responsible for the number of bronchioles or alveoli. But you are responsible for knowing that we have a trachea, bronchi, main bronchus, bronchi that divide into bronchioles. You are also responsible for knowing which of these are in the conducting zone and which of these are in the respiratory zone. So the airways, what's the difference? The difference is in the respiratory zone, gas is exchanged, and in the conducting zone, air is moved, but there's no gas exchange. So the trachea is essential for moving air, but not for doing gas exchange. So the trachea, the bronchi, down to the terminal bronchioles are for moving air, but not for gas exchange, and that puts them in the conducting zone. We have respiratory bronchioles and alveoli, and I don't make a distinction between alveolar ducts and alveoli. Respiratory bronchioles, below the terminal bronchioles, and alveoli that are where gas exchange happens. And so that puts them in the respiratory zone. That's language I do want you to know. So here's a cartoon from your textbook that shows, well, some more anatomical terms. All of these are fair game, either cardiovascular or respiratory. And here is microscopic anatomy, all of which are knowable. I mean, of course they're knowable, they are testable. I could ask you about them. So here is a terminal bronchiole. It's microscopic in size, because this red band is supposed to represent a smooth muscle cell. So here is smooth muscle wrapping, that's the size of a cell, or a couple of cells, and it's wrapping the terminal bronchiole. The terminal bronchiole is not in the respiratory zone, it's in the conducting zone. It has no gas exchange ability. Below the terminal bronchiole is a respiratory bronchiole. So here you divide it into this respiratory bronchiole and this respiratory bronchiole. This one was cut away and we don't see it. Here's a respiratory bronchiole. What makes it respiratory is it's got these little pouches that are surrounded by a network of pulmonary capillaries. And so this is in the respiratory zone because gas exchange happens. That's where gas exchange happens, at the capillary to alveoli or capillary to these pouches on the respiratory bronchiole connection. Alveoli are the dead ends at the end of the respiratory bronchiole that are richly fed with blood, pulmonary artery, a branch. This is microscopic. This is not the pulmonary artery. This is a microscopic arteriole, really, of the pulmonary artery, bringing deoxygenated blood, that's why it's blue, from the right ventricle to the lungs, feeding this rich network of capillaries. And then branches, vein, arteriole, capillary, venule, venule is now red because remember here in the respiratory system, the pulmonary veins are what bring oxygenated blood from the lungs back to the heart. So here is a branch of a pulmonary vein bringing oxygenated blood from the alveoli and from the respiratory bronchioles back to the lung, back to the heart. So this is all, so I'm trying to use the anatomical terms in a physiological context. Are there any questions about this anatomy? Okay, then let's move on, because what we're going to do is move into microscopic anatomy, which is histology. I don't do a lot of histology in this course. It's not a histology course, but I want to expose you to some microscopic anatomy, or histology, the anatomy of cells that are important in a system. So if you go to medical school or nursing school or PA program or maybe even PT, I really don't know, you will probably have a course or part of a course on histology, identifying cells from from photomicrographs, pictures from microscopes. So we're going to be doing some of that, but not a lot. We'll be talking about 5 key cell types in the respiratory system. So just like in the cardiovascular system, where I asked you to think about an SA nodal cell as a cell type, and a ventricular myocyte as a cell type that has a different function from the SA node, or a Purkinje fiber cell. Those, I didn't show you what they look like because they all look kind of the same. The differences are very subtle. So there were cell types. There were SA node cells, there were AV node cells, there were ventricular myocytes, there were Purkinje fiber cells, bundle of His, bundle branch. Those were different cell types. Smooth muscle cells surrounding arteries and veins. Here it gets, in some ways it's easier because the cell types are so more easily identifiable. You can form a picture of a ciliated columnar cell, a ciliated cell. So let's go through some of the cell types. So here is a histological section of a airway. And I think this is a conducting airway. So this is probably, it's a bronchiole, but before the terminal bronchiole. So the airway, let's get oriented, but often if in medical school they won't even give you this orientation. They'll just put up this slide with no labels and you have to identify the cell types. Obviously cells are not purple. The cells are purple and pink because of stains. Yes, and there's a whole art to staining histological sections that we're not going to do, of course. But that's why they're pink and purple. This is the airway. So up here is air or gas. This down here is a basement membrane of connective tissue, but just below this would be blood vessel or some extracellular membrane between the airway and the rest of the body. The point is, so for example, in the trachea, which is surrounded by a very rich sort of rubbery rings of connective tissue, that's down here. And the inside of the airway is lined with these special cells. So we're going to talk about, in the conducting airway, we're going to talk about 2 main cell types. So in this slide, the goblet cell and the ciliated cell are in the conducting airway. The function of a goblet cell is to produce mucus. The function of a ciliated cell, or a ciliated columnar cell, but I just use ciliated because that tells you its function, it makes these cilia and it moves mucus. Production of mucus and movement of mucus is an essential function of the conducting airway, and these are the cells that do it. We're not going to talk about the cells that make connective tissue. We're going to talk about some other cell types later. So how can you tell the difference between them? Well, the ciliated cells have cilia, and the goblet cells have an abundance, like right here, abundance of these secretory granules. You probably, these purple blobs, this blobbiness inside the cell. So this cell is different from this cell right here. That's the cell membrane. And this cell is a goblet cell. This cell is probably a goblet cell. That's because, and it's rich in the production. It has a whole bunch of secretory granules that are where the mucus is secreted from.
How do you know where the ciliated cell stops? Like the hairs on the top and stuff?
Yeah, so this is a surface membrane of a ciliated cell, and these are the cilia that are part of this cell.
But like why is it just like in the middle? Why isn't this cell membrane more to the left and like with all those like the hairs and stuff?
Oh, you mean why isn't it nice and?
Yeah, why isn't it like the whole
There is a whole bunch of them. There's one here. Here is part of another one. Here is part of another one. So this is 1, 2, 3, 4, 5, 6, maybe 7 ciliated cells. Does that answer your question? So they just outlined this one to clarify for you the appearance of the cell. But they could have outlined this one here with the the membrane sort of going down here like this, and then the cilia coming out like that. That's a separate cell. Okay?
Wait, so there's two in that, like one, like where it's outlined, there's one next to it.
1, 2, yeah.
So how can you tell, like I think.
Oh, I won't. Okay. How can they tell? I'm not sure how they can tell, and I sure won't ask you to tell.
Okay.
I'm not going to test you on histology. I'll be honest about it. It's too hard to produce. In black and white, this would just look almost uninterpretable. And I'm not going to print in color. So I do want you to know that there are cell types that you can identify from a color micrograph. And I do want you to know the function of these cell types. But I am not going to put up a slide like this and say which is the columnar cell, the ciliated cell, and which is the goblet cell.
Same with.
If I were to give you a histology question, it would be a cartoon that would be an idealized picture of this, not the real thing.
Same with the type 1 alveolar.
We haven't talked about those, but
Okay,
yes, the same thing.
idea. Okay.
So now we have to talk about mucus. And I forgot to load this up. So let's just, and I haven't looked at this in a whole year, but I remember that these videos are pretty fun to watch. So let me pull this one up. And mucus, you think of mucus, oh, it's gross, and can't we live without it? The answer is no. We can't live without it. We need it. We use it. It's a key function. So mucus is a protein. It's a glycoprotein. It's made from mucins. So mucin is the name of the protein. It's a glycoprotein. Mucin and water make this gooey stuff that is mucus, and we need it. It is a key part of our respiratory system. So let's see what this one is. Okay. So I'm gonna silence it because the sound isn't important. Okay. What am I showing you here? Stop it. This is a primitive vertebrate. It is a hagfish. So it's our evolutionary, great grandparents, right? I mean, we evolved from, we and this hagfish share a common ancestor. This hagfish produces mucus, almost identical to the mucus that we produce in our airway, as a form of protection. So I'm going to dim the lights and show you what this hagfish does. So watch this. So, okay, what are they doing? They are stimulating the hagfish. So this hagfish is anesthetized. They are stimulating the hagfish to simulate an attack on this hagfish. They are collecting the mucus that has been secreted. I'll go back just a few seconds. See? Let's go backwards. Okay, see the white stuff there? They stimulated it, and the hagfish secreted mucus from goblet cells on its skin. They collect this secreted product, and they're just adding it to a glass of water, a beaker of water. So the mucins that were secreted are now being hydrated by the water. So water and mucins make mucus. They're mixing it up. That little bit of secreted mucin, makes a glob of mucus. Okay? You do the same thing, but not quite this robust. So the hagfish evolved to produce mucus as a defense. And here's an interest just to be evolutionary about it. Here's a different video. Okay, well, so there's a hagfish. This one's not anesthetized. It's being attacked. It's producing mucus as a response to the attack. And one more. This one is actually showing some hagfish in their natural habitat. So this is, stop it, that's right there is what I want to see. So this is 8 shark hagfish. Watch what happens when the shark bites the hagfish. It's a mouthful of mucus. In fact, it clogs its respiratory system and can be killed by that clog of mucus. So mucus is our friend, believe it or not. We need it for protection. We don't produce as much mucus as a hagfish, but that's where we evolved this protective mechanism from. So for us, the mucus lines our airway, humidifies the air so that we don't when we breathe in dry air, it gets humidified before it gets into the alveoli. So mucus is important for humidification of the air moisturizing the airway. And finally, we live in a dirty world. We're breathing in dirt and bacteria and viruses all the time. And the mucus traps dirt, bacteria, and viruses, and other things that we don't want. The mucus provides a protective layer for trapping debris, dirt, bacteria, viruses, things we don't want. And then you'll see in a moment, we use the ciliated cells to transport the mucus out of the airway and get rid of the stuff that it's trapped. So here is an electron micrograph that shows a goblet cell. And here's a goblet cell that has just secreted, just dumped some of that dehydrated mucus that will get hydrated with the water that's around it and turn into mucus. So there's a goblet cell caught in the act of secreting. And here are some other goblet cells. Here are ciliated columnar cells, the cilia. And here is what's called a brush cell. And brush cells are the cells that detect irritation. And so brush cells are essential for triggering the cough reflex and the sneeze reflex that you all have experienced. So brush cells are sensory cells that trigger coughing and sneezing as a mechanism of getting rid of whatever it is you just breathed that you don't want. Ciliated cells are cells that move mucus, and goblet cells are the cells that produce mucus. And the cilia, I don't have a good movie of it, but the cilia have a interesting wave-like movement. So they sort of, I'm a ciliated cell, and here are my cilia. They sort of sweep in one direction. The cilia will have a movement like that. They don't sweep back and forth. They do this hula dance, sort of sweep. And so you can see that if there's mucus here, and I'm the ciliated cell in the airway, and I do this movement, I am moving mucus that way. So my lungs are that way. So let's say I'm in the bronchus or something. The lungs are that way. The mouth and nose are that way. I'm sweeping mucus up to the mouth, where the mouth is going to swallow it. So you swallow a lot of mucus every day without even knowing. Your lungs produce it, use it to moisturize and trap debris. Ciliated cells move it, you swallow it and digest it and get rid of it. Yes?
What happens then when we get sick? If all of these three cells work fine, like the bacteria and viruses get trapped and get digested, so what happens when you get like pneumonia, for example?
So pneumonia is a dangerous disease that comes from the production of too much, so there's too much fluid. So fluid actually builds up in the airways where there should not be fluid, there should be air, gas.
Okay.
But even just getting a cold.
Cold, okay.
So in a cold, there is inflammation and irritation of the upper airway, which we have not talked about. But there's ciliated cells and goblet cells there too, and they start producing an excess of mucus. And that can, if you have a sinus infection, same thing. So the excess of mucus is what you have to blow your nose to get rid of. That's excess mucus, that's not, but even now, I mean, as it is right now, there's mucus being produced in your nose that's going down the back of your throat that you may or may not be aware of, that you are swallowing and digesting. Okay, so now we are going to transition from the conducting zone and the mucus production to the airway, to the respiratory zone. And we're going to focus on the alveoli because that's where most of the gas exchange happens. So here's a cartoon of alveoli. So what is this? This is a sort of a sac surrounded by capillaries. Right? There's something on the inside. The white material there is material. It's not just the capillaries. It's white airway surrounded by pink, red, purple, and blue capillary network. So here's what it looks like in an electron micrograph. So here is an alveolus with different branches. So these are the cells that line the alveolus or the alveoli. And because there are so many millions of them, I forgot what I said, 8 million of them or something, and they're all microscopic in size, it produces a very large surface area. So in our lungs, we have a surface area that's about the size of a tennis court. I will not test you on this number. I will not test you on this number. I do want you to know what the alveoli do. Here is another electron micrograph cross-section showing the alveolus surrounded by capillaries. Right? So these are capillaries that have been cut in cross-section, and this is the alveolus that has been cut in cross-section. Air, gas here, blood here. Exchange. That's its job, gas exchange. Here is a cartoon from your textbook that shows this, clump of alveoli filled with air, surrounded by capillaries in cross-section. And now it illustrates the type 1, so type 1 cell and type 2 cell of the alveoli. So this is the air in the alveolus. These are the cells of the alveolus. Type 1 and type 2 cells. There is some membrane interstitium, some connective tissue, very, very thin layer of connective tissue, and then a capillary lined with an endothelium. So endothelial cells, connective tissue, alveolar cells, type 1 and type 2 type of alveolar cells. We're going to talk about the different functions of the type 1 and type 2 cells in a minute. This is all microscopic. So here, this illustrates this very well. This is a red blood cell. So it's going down into the subbasement, it's going, out the building. This is just a piece of a red blood cell. Here's the membrane of a red blood cell. Here's the type 1 alveolar cell. Here is the basement membrane, the connective tissue between the alveolar cell and the endothelial cell of the capillary. So this is red blood cell, this is blood plasma, endothelium, connective tissue, type 1 alveolar cell.
Now, on the previous slide, do you get 2 blood cells and 1 alveolar?
See, this is just a cartoon.
Oh, you can get more than 2?
No, I mean, you can see, but the point of this, there might be more than 2, there might only be 1. The point here is that a capillary has a diameter that's about the same size as a red blood cell. The red blood cell and the capillary are approximately the same size.
Yeah, so last week you said that only 1 blood cell can pass through a capillary?
Yeah, so this is a cartoon that shows 2. 1, 2. Is that okay? I mean, typically,
Yes, I
it's,
think I
so,
understand what it's saying, and maybe like we have more than 2 blood cells.
no,
in one.
no, let's just say it's 1. Let's say this is a artistic license, but there's 1 red blood cell going through a capillary at a time. It's just easier to, I don't know why the artist put in 2, if So that's a good point. If I had been editing this book, I would have said, let's just make it 1, because that's more realistic. I mean, that red blood cell looks like a red blood cell. You could read it one at a time. Other questions about this slide? So this is the alveolus, and this is the blow-up of just this one section of alveolus. I didn't say that before, but now I have. This is the business end of the airway. Air, type 1 cells, capillaries. Here the cartoon is showing that there is cilia in the respiratory bronchiole. Maybe, maybe not. We don't produce mucus in the respiratory bronchiole. We produce mucus in the conducting bronchiole. So I think, I'm not convinced that this cartooning of cilia at the junction between the respiratory bronchiole and the alveolar sac is accurate. But the point, but the point is useful, that to remember that the bronchioles are where cilia are, and there are no cilia in the alveoli, and there should be no mucus here either. We've moved the mucus, any mucus that was produced, let's just say any mucus that was produced near here was transported by the cilia that way. This should be dry. I mean, not dry, it should be air, not fluid. So you can see this microscopically thin layer between air and blood. And that facilitates gas exchange because the law for diffusion goes like this. And I'm not going to ask you to work with this, but just point out that the flow due to diffusion is proportional to the gradient of concentration. It's inversely proportional to the thickness that it's diffusing through. And it is proportional to the area that it has to diffuse through. So the higher the area, the more diffusion. And that's why we have a tennis court of surface area in our lungs. High area. The thickness is extremely thin. Very thin, it means efficient diffusion. So we have evolved a mechanism to allow diffusion, just simple diffusion. The gases dissolve in fluid and in the membrane. Simple diffusion, we talked about this a few weeks ago, is sufficient for our gas exchange. We do not have active transporters for oxygen or carbon dioxide. We simply use simple diffusion. And that's facilitated by a high area and thin thickness. The rest of this stuff we don't need to know about. So what I would like you to do now, I think now, let me just double check before I move forward. Yeah, what I would like to do now is to take a break and for you to work on the clickers that follow. So you have a bunch of clickers. I mean, there's 4 slides, but there's actually 8 questions, because each slide has 2 questions. Work on the clickers, either individually or together, safely. I'll go through the answers quickly at the beginning of the next section. So let's take, 10 or 15 minutes as a break, stretch, relax, do the clickers. We'll come back and do them together and start the next section.